Hello guys, this is a video of Franklin OKEY-IWOBI and Danie Martinez, and this is our transcription project. So first, transcription and translation is how we go from DNA to RNA to proteins, which makes us from our food and all of that. And it's called the central dogma, which is basically the way of life, which is how we live, how we make the proteins that we need to survive. Alright. And this first video is going to be about transcription. So transcription is when the genes in the DNA are copied into the mRNA, which then leaves the nucleus. So it basically copies down the code that were in the DNA and sends it somewhere else to make proteins. Alright. So this is the nucleus. Inside the nucleus, there's the DNA and the DNA. It has two different strands. The first one is runs from 3' to 5', called the antisense strand or the template strand. This strand right here, and this is what the mRNA. This is what the polymerase copies off, and then this is the sense strand, which doesn't code, but it has the genes, similar genes that the mRNA has. So what happens is that the DNA polymerase starts the transcription process by breaking apart the strands of DNA, and then after it does that, it brings in the different mRNA strands and matches it with its complementary pair. So thymine goes with adenine, adenine goes with uracil. So instead of thymine, like a regular DNA process, RNA has uracil instead of thymine. And then cytosine goes with guanine, and the rest go together. And after that, it breaks this apart and it pairs up the mRNA. And after it's done coding that, the mRNA leaves the nucleus after it's been done copied. And before it leaves the nucleus, there are some parts of the mRNA called the introns that don't code for anything particularly. So it could be a switch off and off gene. Then the mRNA gets rid of that before it leaves the nucleus, and it stays inside the nucleus. And then the coding part leaves the nucleus into the cytosine to go find the ribosome where it translates later. But then the other strand of DNA attaches back to the DNA through its hydrogen bonds that were there originally, and then the DNA is almost exactly as it was before the transcription process happened, so it's all together. That's the DNA, and this was the mRNA.